I'm happy to introduce Dr. Kristin Bianchini, who's joining us today. She got her undergraduate degree in physiology and developmental biology from the University of Alberta, a master's degree in integrated biology from the University of Guelph, a PhD in ecotoxicology from the University of Saskatchewan, and she's done postdocs with Acadia University and Birds Canada, and she's with Birds Canada now as a waterbirds and wetlands scientist. And so I'm thrilled you can make it with us today. We kind of met, well, we haven't actually met. We exchanged emails when she was working on this project that she's going to talk about today. I got an email with she was looking for folks to help collect sediment samples so she can test them for these pollutants. So welcome and thanks for joining us today.
Yeah, thank you. Thanks for having me. All right, so let's get started. Okay, yeah, so thank you all for tuning into my talk. Today I'm going to present some work that I completed as a part of my PhD at the University of Saskatchewan with Dr. Kristin Morrissey. And it's looking at how oil spill pollutants impact shorebird fueling and migration. The first point that I want to emphasize for this talk is just how amazing bird migration is. So I was super lucky to be able to study bird migration during my PhD. And I think that one of the biggest takeaways of my research was a huge appreciation for how incredible avian migration is. So for anyone who isn't already familiar with birds, I'm going to walk through a few of my favorite examples of just how impressive bird migration is. Specifically for this talk, I'm going to focus on shorebirds. So these are the small to large birds that you'll usually see wading in the water or running up and down the sand along the beach. They're usually kind of brownish in color, and they can also have black or red markings. And where you guys are in Louisiana, you'll usually see shorebirds on the beach throughout the winter and in the early spring and fall. The first example of shorebird migration that I'm going to talk about comes from this little shorebird on the right of this picture. This bird is called the semi-palmated sandpiper. So semi-palmated sandpipers weigh about 22 grams on average, or about 0.05 pounds. Now on one leg of their migration, these small birds will make a non-stop flight of 5,300 kilometers, or about 3,300 miles, between Canada and South America. So for any runners out there, these small birds are essentially doing 126 marathons in a row. Now I should mention that because these birds are flying, they're essentially sprinting these 126 marathons without stopping to rest for food or for water. The most extreme example of shorebird migration is the bar-tailed godwit. These birds will make a non-stop flight from Alaska to New Zealand. In total, they fly non-stop for about 12,000 kilometers or 7,500 miles. This trip takes godwits about 6 to 9 days, and it's one of the longest known non-stop flights in shorebirds. But not all birds complete their entire migration in one giant flight. Instead, most birds stop along the way to refuel. So an example of this is a bird that I worked with for my PhD called the red knot. Red knots are still long-distance migrants, so they make non-stop flights of thousands of miles as they travel from their wintering grounds in South America to their breeding grounds in the Canadian Arctic. So unlike the bar-tailed godwit that I showed before, red knots will stop at specific sites along their migration route to rest and to refuel. These sites are called staging sites. And the process of refueling is called pre-migratory fueling. So pre-migratory fueling is a period of intense food consumption called hyperphagia, where birds eat a lot of food. And they store all of the food that they eat as fat. Now, shorebirds are extreme fuelers, and they're capable of more than doubling their body mass at a single stopover site. Now, all of the fat that birds accumulate while they're at their staging areas is their only source of fuel during these long-distance migration flights. For this reason, proper fueling is really important for long-distance migratory shorebirds. It increases the probability that birds survive migration because it makes sure that they have enough fat to reach their next staging area. And proper fueling also increases a bird's reproductive success because the faster that a bird puts on fat, the faster that it migrates. And birds that reach their breeding grounds first have access to the best breeding territories and have higher reproductive success. So in this way, storing enough fuel and rapidly storing enough fuel is really important for these birds. So for this reason, anything that impairs fat accumulation can have really negative consequences for migratory birds. And as a result, lower refueling rates and lower departure masses have been associated with population declines in long-distance migratory shorebirds. So for example, a lot of work has looked at the relationship between impaired fueling and population declines in red knots. Research by Baker and colleagues looked at red knots at a staging area in Delaware Bay, New Jersey, and they found that over time, the departure mass of red knots was decreasing. So this means that red knots were putting on less fat than they did in the past. Because of these declines in departure body mass, annual adult survival had declined by 37%. And this population of red knots had decreased from 51,000 to 27,000 birds on their wintering grounds. And demographic modeling predicted that if this trend continued, this population of red knots would run the risk of extinction. So this study really emphasizes that pre-migratory fueling is really important for long-distance migratory shorebirds. Now, as a whole, impaired fueling is a big problem for shorebirds because they're currently experiencing high levels of population decline. This figure is showing the percent change in populations of different bird guilds in Canada from 1970 to 2016. Shorebirds are shown in orange here, and you can see that since 1970, their populations have declined by about 40%. And it's thought that one of the factors contributing to these declines is impaired pre-migratory fueling. There are a lot of different factors that can impair pre-migratory fueling. Reduced prey availability is an important factor when we think about fueling, because if you can imagine at a site where there's less food for birds, they don't have the source of fuel that they need to put on their fat. Human-related disturbances are also a big problem for shorebirds. A lot of beaches can be really crowded with people, and so people just take up a lot of space. And that means that any space where there are people are spaces that shorebirds can't access to get the fuel that they need for migration. High predator presence can also affect fueling, but it's actually been found that shorebirds time their migration so that it doesn't overlap with the migration of raptors, which are shorebird predators. Reduced habitat quality and habitat loss is an especially big problem for shorebirds, and this is because they rely on specific sites to refuel. And so the loss of those areas means that sites that they use to refuel don't exist anymore, or the quality of those sites is a lot lower. And finally, stopover site contamination is a really big problem for shorebirds. And stopover site contamination is what I was looking at for my PhD research. And specifically, I was studying how oil spill pollution can impact pre-migratory fueling. So as I'm sure most of you are aware, oil spills are a major environmental issue. It's been estimated that over 4.5 million tons of oil have been spilt since 1967, and that about 170,000 tons of oil were spilt per year between 1991 and 2012. Oil spills are a long-term problem for birds because oil pollution can persist for decades following an oil spill. So for example, 12 years after the Exxon Valdez oil spill, 100 tons of oil were still found in intertidal subsurface sediments. Often this oil wasn't weathered because different physical barriers had prevented things like disturbance, oxygenation, or photolysis from degrading the oil. Also, 16 years after the Exxon Valdez oil spill, polycyclic aromatic hydrocarbons, or PAHs, were elevated in subsurface sediments. Now, polycyclic aromatic hydrocarbons, or PAHs, are considered the toxic components in oil spill. And this is what my work focused on. So as I said before, PAHs are the primary contaminant of concern in oil spills. They can persist in the environment even after the oil itself is no longer visible. PAHs can bioaccumulate in marine invertebrates. So this means that they can reach higher and higher concentrations over time. And PAHs also have the potential to be incorporated into the food chain. So this means that they can be taken up by higher trophic levels like birds. So taken together, this means that PAHs pose an important chronic risk for birds following an oil spill. Because of shorebirds' life history, they may be particularly vulnerable to chronic PAH exposure. Just the act of hyperphagia, where birds eat a lot of food at a certain site, means that shorebirds can have a high risk of contaminant uptake if they're refueling in a contaminated area. Shorebirds also have really high staging site fidelity, which means that they tend to return to the same sites every year during migration. And so if they're returning to a contaminated site every year, that means that they're being exposed to those same contaminants over and over again. And finally, these long-distance migrants operate within a very finite physiological window. So even small changes in fueling can have really negative consequences on the shorebird's survival and reproductive success. So the reason that I was looking at PAHs and fueling is because previous research suggests that PAHs have the potential to impair pre-migratory fueling. So previous work has found that PAHs are negatively associated with body condition in birds, meaning that at higher levels of PAHs, birds have less body fat. PAHs can also cause thermal stress in birds, and birds often compensate for elevated heat loss by increasing their metabolic rate, which causes them to lose mass. PAHs can also cause liver and intestinal damage. Damage to the liver would impair fatty acid synthesis, and damage to the intestine would impair the absorption of food. And PAHs can also disrupt the signaling of estrogen, thyroid hormone, prolactin, and corticosterone, which are all hormones that are involved in the endocrine control of pre-migratory fueling. And finally, PAHs can also bind to the aryl hydrocarbon receptor. This is an important receptor for a class of contaminants called dioxin-like compounds. Dioxins were a really big problem decades ago, so a lot of research has looked at how dioxins impact animals. Dioxins exert most of their toxic effects by binding to the aryl hydrocarbon receptor. So because PAHs also bind to this receptor, they can have effects that are similar to dioxin-like compounds. So to show these effects, I'm showing the cycling of fatty acids between the liver, where fatty acids are produced, the adipose tissue, where fatty acids are stored, and the muscle, where fatty acids are oxidized to produce energy. So previous work has shown that activation of the aryl hydrocarbon receptor can inhibit fatty acid synthesis and lipid export from the liver. It can inhibit the differentiation of pre-adipocytes into mature fat cells. It can inhibit the enzyme lipoprotein lipase, which is involved in fatty acid uptake. It can increase peripheral fat mobilization from the adipose, or increase peripheral fat mobilization from adipose tissues, and it can cause hepatic steatosis, which is a form of liver damage. So there's a lot of evidence that shorebirds have a high risk of exposure to PAHs, and there's also a lot of evidence that PAHs have the potential to impair pre-migratory fueling. But despite all of this evidence, we had yet to actually establish a link between exposure to these compounds and impaired pre-migratory fueling. So this brings me to the objectives of my research. So my first objective was to directly test whether PAHs affect avian pre-migratory fueling. If I found that PAHs did affect fueling, then my next objective was to look at what mechanisms could be causing this fueling impairment. And finally, my last objective was to examine the relationship between PAH exposure, fueling, and migration timing. I accomplished my first two objectives using a captive dosing study. For all of the research that I'm going to talk about today, I use Sanderling as a model. Sanderling are long-distance migratory shorebirds. They're capable of extreme pre-migratory mass gains. So for example, at their staging area in Delaware Bay, they can increase their body mass by over 70%. And Sanderling also feed on along intertidal shoreline habitats, and they have a subsurface probing behavior, which makes them have a really high risk of oil exposure. So all these characteristics make Sanderling a really ecologically relevant study species for looking at how PAHs impact pre-migratory fueling. So for my captive dosing study, I brought 49 Sanderling into captivity. When the birds started to exhibit signs of pre-migratory fueling, they were split into a control, low, medium, and high dose group. These birds were dosed with either organic sunflower oil or with 12.6, 126, or 1260 micrograms of PAH per kilogram body weight per day. I chose these concentrations because they were measured previously as dietary pH exposures for Sanderling following an oil spill. And the mixture that I used for dosing was a commercial mixture of PAHs that had previously been measured following an oil spill. This mixture contained 15 of the US EPA's 16 priority pH compounds. To dose the Sanderling, I took my dosing solution and injected it into a mealworm, and then I fed that mealworm to the Sanderling. So this mimicked the ingestion of a contaminated food source during staging. For my dosing experiment, I dosed the birds every day for 21 days, which is the average amount of time that Sanderlings spend fueling at a staging area before they depart for migration. So this was also an environmentally relevant period of exposure. Throughout the experiment, I took daily mass and body morphometric measurements in order to measure changes in fuel loads over time. And then at the end of the experiment, I collected different samples from the birds. I took a blood sample in order to look at different biochemical parameters in the Sanderling serum. The parameters that I looked at were indicative of the bird's fueling status, of liver, kidney, and muscle damage, and of lipid homeostasis. I also took liver samples from the birds, and I used these to perform an EROD assay to look for AHR activation and PAH exposure. And I also did real-time qPCR on my liver samples to look at changes in hepatic mRNA expression. The first result of the captive dosing study was looking at changes in Sanderling body mass over time. So I found that in birds dosed with PAHs, they gained less mass over time than the control birds. So this result confirmed that PAHs lowered Sanderling pre-migratory mass gains. And so my next objective was to see what mechanisms could be driving this result. To answer this question, I looked at my gene expression and blood biochemistry results. So CYP1A4 and EROD activity are both markers of aryl hydrocarbon receptor activation and PAH exposure. And I found that CYP1A4 and EROD were both elevated in the high dose group relative to controls. So these results confirmed that the aryl hydrocarbon receptor was activated, and it also confirmed that the birds were actually exposed to PAHs during the dosing experiment. So next, my gene expression results also showed that in the high dose group, liver fatty acid binding protein and lipase expression were reduced relative to controls. Now, I'm also showing analytes, bile acid analyte in the serum. Now, for all of the different compounds measured in the serum, I actually found that EROD activity was a better marker of exposure than the identity of a bird's dose group. And this was probably due to differences in metabolism or gut absorption efficiency among the individual birds. This is why you see EROD activity on the x-axis here. So I found that with higher EROD activity or with higher levels of PAH exposure, bile acid concentrations in the serum were lower. So fatty acid binding protein is an intracellular lipid chaperone. It enhances fatty acid uptake, storage, transport, and metabolism. Hepatic lipase is an enzyme that breaks down fats to enhance their absorption. And bile acids emulsify lipids, so they help with fat digestion and absorption. So together, these results suggest that PAH exposure lowered Sanderling's ability to transport, metabolize, and absorb lipids. So going back to my objectives, I confirmed that PAH exposure reduced Sanderling pre-migratory mass gains. And my results also suggested that PAHs interfered with lipid homeostasis in these birds. So this brought me to my third objective, which was to examine the relationship between PAH exposure, fueling, and migration timing. And I accomplished my third objective using a field study. For my field work, I trapped birds during spring migration at 3 sites in the Gulf of Mexico. So I trapped birds from North Padre, Texas, from Bolivar Flats, Texas, and from 3 sites in Louisiana. So I was at Port Fourchon, Grand Isle, and Elmer's Island, Louisiana. I chose these sites for my field work because they were variably affected by the Deepwater Horizon oil spill, and they're also sites that are prone to frequent and repeated oil pollution. So at each site, I measured birds' fuel loads by taking mass and body morphometric measurements. And I also measured each bird's fueling status by taking a small blood sample to measure plasma metabolite levels. I also attached coded nanotags to a subset of birds. So these are really small tags that weigh less than a gram, and I glued them to the bird's back under its feathers. Now, these tags transmit a signal that can be detected by these receivers. The receivers that I was using were part of the Modus wildlife tracking system. So this is a network of receivers that's located throughout North America. And this allowed me to track shorebird migration so that I could look at departure dates, stopover durations, and travel times between different sites. And finally, I also took sediment samples from all of the sites where I trapped the birds to look at PAH concentrations. And this allowed me to estimate shorebird PAH exposure during foraging. So the first result from my fieldwork was the sediment PAH concentrations. So on average, sites in North Padre had PAH concentrations of about 30 nanograms per gram. Sites in Bolivar Flats had really similar PAH concentrations. But then at one site in Louisiana, at Elmer's Island, PAH concentrations in sediment were over 600 nanograms per gram. Now, from my radio tracking data, I found that birds that were trapped in the North Padre staging area tended to stay in North Padre. Birds that were trapped in Bolivar Flats tended to stay in Bolivar Flats. And the birds that were trapped at 3 sites in Louisiana tended to stay at these 3 sites. So there wasn't a lot of movement between these 3 staging areas. So because Louisiana had high PAH concentrations and the birds tended to stick around in this area, it suggests that Sanderling that were refueling in Louisiana were exposed to higher PAH concentrations than birds from the other 2 sites. So next I looked at differences in fueling between these 3 areas. To measure fueling, I used my plasma metabolite data to calculate a fattening index. So a higher fattening index indicates that birds had higher fueling, and a lower fattening index indicates that birds had lower refueling. So I found that Sanderling in Louisiana, where there were higher PAH concentrations, were in a more catabolic state. So they had the lowest fueling potential. But despite fueling more slowly, Sanderling that were trapped in Louisiana departed at the same time as birds from Bolivar Flats. So this is a little bit unexpected. Now there are 3 possibilities that we think could be explaining these results. The first possibility is that because Sanderling and Louisiana were refueling more slowly, they could be departing with less fat. But unfortunately, we don't know this for sure because we weren't able to retrap the birds, so we don't know their departure mass. Another possibility is that other factors that we weren't measuring here, like differences in weather or differences in food quality between these three sites, was affecting Sanderling departure dates. And the third possibility is that Bolivar Flats might also have had high contaminant levels. So Bolivar Flats is located in the Houston Ship Channel, which is known to be heavily polluted with municipal and industrial discharges. So there could have been other contaminants other than PAHs that were influencing departure dates in this area. So nevertheless, I did find that there was a negative relationship between mass at capture and departure dates from the Gulf of Mexico at all three sites. So this suggested that fuel loads did impact departure dates because heavier birds tended to depart earlier. So my next questions were, how did departure dates affect a bird's subsequent migration pace? And essentially, did slower birds catch up? To answer these questions, I looked at all of my radiotelemetry detections north of the Gulf of Mexico. So 35 of the Sanderling that I tagged in the Gulf were detected north of this area. So of these birds, 2 were detected flying north along the Atlantic flyway. And the remaining 33 birds, or 94%, were detected in Saskatchewan, Canada. So there was really strong connectivity between the Gulf of Mexico and Saskatchewan. And for these results, it's important to note the distribution of MODIS receivers throughout North America. So there's really high coverage along the Atlantic flyway, but there are very few MODIS receivers in the mid-continent. So because of the high coverage along the Atlantic flyway, we would expect that any birds that were detected or that flew north along this route would be detected. So the fact that most of our birds were detected in Saskatchewan really speaks to the importance of this interior route for Sanderling. Now, I also want to note that because there's such low MODIS coverage in the North American interior, we had really low recovery rates for our tags. Only about 29% of the birds that we tagged in the Gulf of Mexico were later detected at north or more northern staging areas. So this suggests that birds were likely stopping over at other sites in the interior where there were no MODIS receivers. So for Sanderling that were detected in the Gulf of Mexico and in Saskatchewan, I was able to look at the timing of migration as birds traveled between these two areas. So if you remember from the results that I showed before, Sanderling from Louisiana and Bolivar Flats departed later than Sanderling from North Padre. Now we expected that birds that departed later from the Gulf would arrive later in Saskatchewan. But unfortunately, the story got a bit more complicated after birds departed from the Gulf. And this was because Sanderling from North Padre showed some really unexpected results with respect to their migration timing. So Sanderling from North Padre departed earlier than birds from the other two sites. So we would expect them to arrive earlier in Saskatchewan. But instead, we found that North Padre Sanderling traveled more slowly to Saskatchewan. And because they traveled more slowly, North Padre Sanderling arrived in Saskatchewan later than birds from the other two sites. So next, we looked at what was happening in Saskatchewan. We found that even though North Padre Sanderling arrived later, they spent a lot less time refueling. So they had shorter staging durations in Saskatchewan. And because of this, Sanderling from North Padre departed at the same time as birds from the other two staging areas. So there's no difference in Saskatchewan departure times among birds from these three areas. So these migration results are a bit confusing. These lower travel times suggest that Sanderling might have stopped over at unknown sites on their way to Saskatchewan to refuel instead of making a direct flight. This would imply that the birds were not time constrained, so essentially that they were in no rush to reach their breeding grounds. But we think that this conclusion is unlikely based on what we know about shorebird ecology and spring migration strategies. So we think it's more likely that Sanderling and North Padre were traveling outside of the detection range of MODIS receivers at the site before actually departing for migration. Oops, sorry. Okay. On the other hand, the shorter staging durations in Saskatchewan suggest that Sanderling were refueling faster at the site in order to reach their breeding grounds as early as possible. So this suggests the opposite, that Sanderling were time constrained and they were trying to reach their breeding grounds fast. And this agrees with our understanding of shorebird ecology. So we have some conflicting results here from my research, and more work is needed to really dive into what's happening with these birds and why we're seeing these differences between the three Gulf of Mexico staging areas. So two things that I want to note are that we did have really small sample sizes for birds that were detected in both the Gulf of Mexico and in Saskatchewan. So since I finished working on this project, there's been a lot more work to put radio tags on shorebirds and to install MODIS receivers in the North American mid-continental flyway. So hopefully the combination of more tags and more receivers will provide more information to help us understand whether there are actually carryover effects from staging areas in the Gulf of Mexico. So coming back to my third objective, I found that sediments in Louisiana had the highest pH concentrations. I found that fueling rates for Sanderling were lower in Louisiana, and that lower fuel loads were associated with later departures. But more research is needed to investigate the impacts of later departures on subsequent migration timing. So as you can see, after finishing my captive study and my field study, I have a lot of data about how PAH exposure impacts Sanderling physiology and migration timing. So my next step was to see how are all of these data linked. So the last goal of my PhD was to put together a possible toxicity pathway showing how PAH exposure impacts shorebirds. For my work, I collected data that spanned from the molecular to the individual level. My captive study showed that PAH exposure lowered Sanderling pre-migratory mass gains, and it suggested that a possible pathway for this impairment was activation of the aryl hydrocarbon receptor and the disruption of lipid homeostasis. So we can tentatively link these different effects together in a possible pathway for pre-migratory fueling impairment. But I want to note there are a lot of different pathways that regulate pre-migratory fueling. So more research is needed to see if this is actually the pathway that's involved here, or if there are other pathways that are involved. Now, specifically, we have to determine if these effects are causal. So if AHR activation is actually causing a disruption in lipid homeostasis, and if this disruption in lipid homeostasis is actually what's leading to a decrease in pre-migratory mass gains. So more research is needed to link these molecular to the individual level effects. But despite the pathway involved, my work provided pretty clear evidence that PAH exposure impaired Sanderling pre-migratory fueling. There's also evidence from my fieldwork that birds that had lower fueling had later departures, which suggests that PAHs have the potential to impact migration pace. But it's unclear from my work whether delays in departure carried over to affect birds at more northern staging areas. But even though I didn't see clear evidence that impaired fueling caused migration delays in my work, there's really strong support for this relationship from the literature, where previous studies have shown that at sites with less food or with lower quality food, birds don't fuel as quickly. And because of this, they migrate more slowly. There's also a lot of support in the literature linking lower fueling and migration delays to population level impacts. So for example, there's work linking impaired fueling to later arrival on the breeding grounds, to lower reproductive performance and lower survival, and then to population declines. So the evidence linking lower fueling to population level effects is pretty clear in other bird species. So this is the proposed toxicity pathway linking PAH exposure to impaired fueling, delays in migration, and population declines. More work will be needed to link together these molecular to individual level effects. More work will also be needed to see if these effects, especially from the individual to the population level, are also true for Sanderling. And we also want to see if this proposed pathway or similar pathway could be more broadly applied to other bird species, and also to see if this pathway can be applied to other chemicals with similar mechanisms of action. To summarize, from my work, I found that PAH exposure can impair pre-migratory fueling and that it has the potential to impact migration pace. And more work is needed to really tease apart this toxicity pathway linking PAH exposure to impaired fueling and its impacts on migratory shorebird populations. Understanding these toxicity pathways is really important because it helps us to understand how PAHs and compounds with similar mechanisms of action affect bird populations. And these pathways also help to inform avian ecotoxicological risk assessments. And ultimately, this helps to improve the conservation of long-distance migratory birds, which is the main reason why we do all this research. So I'd like to thank everybody who helped me with this research. So my PhD advisor, Dr. Kristin Morrissey, all of the committee members for my PhD, Lori Wilson and the Chaplin Nature Center in Saskatchewan, David Newstead in Texas with Texas Coastal Bends, Bays, and Estuaries Program, Delana LeBlanc and the Barataria Terrebonne National Estuary Program in Louisiana. I'd like to thank everyone who helped me in the field and in my captive work. I'd also like to thank all of the institutions and researchers that contribute to the MODIS network and the landowners in Saskatchewan who let us put MODIS towers on their land. I'd also like to thank NSERC, the University of Saskatchewan, and Environment and Climate Change Canada for funding. And I would be happy to take any questions.
Great, thanks. That was super interesting.
Thanks.
I don't see any questions in here yet, so y'all go ahead and type in. Oh, okay. Did you test for general differences in food availability among Gulf Coast sites?
No, unfortunately, we couldn't look at, or we didn't look at food availability, but that would be a really interesting factor to look at. There would be a lot of different habitat differences between these sites, and those could definitely be affecting the birds. So that would be a really interesting area for research.
Okay. Well, I got a question. So when these birds are fueling up, getting ready for migration, they not only put on fat, there's some evidence that their muscles actually become more, for lack of a better word, fit. So it's almost like they're exercising without exercising. So their muscles get ready for it without, which is really, really interesting to me, without actually, working those muscles. So do you know if there's any evidence that, exposure to contaminants like this affects that process? Because muscle's heavier than fat.
Yeah, that's a really good question, and I didn't show it in this talk because it doesn't really relate to the whole like fat story. But one of the results that I did find is that creatine kinase in the blood decreased, or wait, oh no, it increased. Yeah, so it showed that there was muscle damage, and we don't know where that muscle damage was coming from, but that would be a really interesting follow-up to the study to see, like, are the muscles being affected? Because that would definitely affect the birds. They wouldn't be able to fly as well, and yeah, they wouldn't reach their next sites as easily.
Okay, so were there body quality differences between birds that had to fly fast to Canada and those that flew slow after they arrived in Canada?
Oh, so differences in body condition between the three sites, I guess. Yeah, we didn't, we only saw differences in the plasma metabolites. So that's like an indication of how quickly birds are putting on fat. It's hard to really tease apart what the fat level would be because we trap birds at different times. Like you can imagine we're driving from one site to another. So the last site that we reach, birds are going to automatically be fatter than at birds at the first site that we go to. And so We account for that statistically by just including trapping date as a random effect in our models. But that's why I really looked at the plasma metabolites rather than body condition as a measure of how quickly birds are putting on fat.
Are there any known ways to decrease PAH as of right now?
That's a good question. I'm not sure if there's a way to kind of remediate the environment from PAHs. I'm sure that people would be very interested in that research if it were possible, but I'm not sure because the PAHs just kind of get into sediments and tend to stay there. They degrade over time, and light exposure, for example, will degrade PAHs and cause them to break down. But it's just when they get trapped below the surface in the sediment, they don't really break down as quickly. So that would be tough. And yeah, it'd be interesting to know how they do that. But that would be more kind of an engineering problem, I think.
Yeah, so it might be possible if you're doing some restoration where you're pulling up sediment and depositing it to build an island, or say that it could release a lot of these PAHs that are trapped, but perhaps make it exposed to the, you know, to light, which could help break it down. So
yeah.
Is that a good thing? You know?
I think maybe it depends on when the birds get there in relation to when you do that, maybe.
Yeah, yeah.
Yeah.
Could we look at the results page again, the one with your questions and the results found?
Okay. The objectives, I guess. This one.
Amelia, let us know when we get to the slide you want. This one, okay.
Okay.
Did you have a question about it? You just wanted to see how pretty it was again. Oh, okay. Just couldn't read it. Okay.
Okay.
Wanted to read it again. Does anybody else have questions? It's funny because I started thinking, because Dr. Newman does a lot of research about adipose and development of adipose from stem cells. And it doesn't seem like a very good pathway, but you know, I started thinking about it from the point of obesity, right? If you could interrupt this pathway that perhaps not in a way that damages like is done here, that could be an interesting, just interesting to understand the pathway.
Yeah, that is interesting.
Okay, here's one. In addition to the impact of pH on shorebird fueling, could the presence of environmental stressors, I gotta scroll down and it's not scrolling, okay, such as close proximity to heavily industrialized areas like the Houston shipping channels, also contribute to impaired fueling in the birds?
Yeah, absolutely. And so that goes back to that slide where I showed that multiple different factors affect fueling. And so, yeah, it's really important to remember that PAHs aren't the only contaminant, and it isn't the only stressor that these birds are being exposed to. It's just multiple factors all affecting them all at once. And I think more research is trying to move into looking at whether these effects are synergistic and how all of these effects kind of interplay and affect the birds all at once. And yeah, there's also, like, these birds are visiting multiple locations. So for migrants, it's really difficult to tease apart how all of the different effects at all of the different sites are affecting an individual, but they're definitely being affected by lots of factors.
Mm-hmm. You mentioned how the oil pollutants get into the food chain, into the fish, and then the birds. Does that mean the PAHs can be in the fish we eat?
Well, for PAHs, they accumulate in marine invertebrates, but a lot of vertebrates break down pHs really well. So it's not necessarily a contaminant that really biomagnifies, like with mercury, for example. Mercury definitely moves up the food chain, and the higher that you get, like in higher trophic levels, they're going to have higher concentrations. But pHs don't completely work the same way. So Sanderling are definitely getting this from the invertebrates that they ingest and from sediments that they ingest just while they're eating. But I think I'm not familiar with fish physiology, but I would assume that they would probably break down pHs in the same way that birds would. So you probably don't have to worry about being exposed to pHs as much, but there are definitely other contaminants that will biomagnify. But PAHs aren't necessarily one of them.
So was your field work, was it done in 1 year, or was it over multiple years?
It was 3 years.
3 years.
And then I had 4 years in Saskatchewan.
Okay. So the data you showed us was all the years combined?
Yes, it was.
Yeah. Okay.
Yeah, so we had 120 tags on all the Sanderling total. Was there.
Oh, wow. Okay. So then you only got data from 33 of that 129. Oh, wow.
Yeah. Mm-hmm.
Wow. Okay. So folks have been working on this since you finished with it. Do you have any preliminary information for us that you could share?
I don't. I know what they're doing, but I'm not sure what they've found yet.
Okay.
So I know the PhD student that followed me, their project sounds really incredible. They're going down to South America and looking at the wintering grounds and shorebirds on their wintering grounds and seeing what kind of contaminants and different things they might be picking up there, and then going to the Gulf as well, and then in Saskatchewan. So trying to link kind of all of these 3 stages of their migration together.
Oh, wow.
So it should be pretty interesting. And they're also using isotopes to look at kind of food and trophic levels at the different locations. So it's a really cool project, but I'm not sure what they found yet, unfortunately.
Okay. It will be interesting to keep posted on that.
Mm-hmm.
Anything else from anybody? Oh, I missed one question. Would oil pollution negatively affect the growth size of fish or birds?
Oh, like the developmental growth? Is that?
Sure.
Yeah, so for
Yes.
for my project, I only looked at birds that were so they're called after second year birds. So second year birds means that they're on their first migration north. And we didn't look at those birds just because they tend to have a different migration schedule than adult birds, and they also don't fuel the same way. And I'm not sure if they presumably should be done growing at that time. So it wouldn't necessarily affect their growth. Like they do most of that on their breeding grounds before they migrate south. So the adults, they would already have grown. So it's more just affecting like their ability to put on fat. I'm not sure about fish. That's a good question, because the fish, well, most of the fish would probably be in that same area and be developing over time, but I'm not sure how it would affect fish, but that's a good question.
Well, so ingestion of oil compounds can affect how the gut can absorb nutrients, right? So presumably, if these young individuals are being fed contaminated foodstuffs, that it could interrupt the absorption of nutrients, which just logically would link it towards impaired growth. So that could happen. I don't have any data to cite for that, unfortunately.
Yeah, it makes sense that it would, because PAHs do cause intestinal damage, so it would affect the whole process. But yeah, I also don't have any data
Yeah.
for that.
Be nice to have that data.
Yeah, for sure.
Anything else? All right. Coming up on the last chance to get your question in. All righty. Well, I guess that's it. So thanks so much for spending your time with us and sharing your really super awesome research with us.
Yeah, thanks a lot for having me. Yeah, it was great to share it.
Yeah. All right.